So, first of all, I would like to thank organizers to giving me an opportunity to present my work today. And yes, it's a short talk, so we will be doing this briefly. As I said, from Antarctica, we are coming to the Dead Sea. Hope I can move page down. Oh, yes. So from the Antarctica, we are coming to Dead Sea. Is in Israel, we're situated under the level of the sea, almost 500 meters under the sea level. And the story starts already 40 years ago when the professor from the Haifa University in his mycology lab and the famous professor Aviatar Nevo, what have been working in evolution, evolutionary career, found together with the professor Solomon Wasser the fungi, Penicillium fungi, in the near or close to Dead Sea, and we received the sample, this new organism, and we were asked to annotate and study this organism. So we ran DNA RNA sequencing, extraction sequencing, and we tried to study and find new genes responsible for high salinity, osmolarity, and different bioactives molecules like penicillin production. And we ran the in silico comparative study with other organisms. So in principle, this is the organism in the central image found here in the Dead Sea, and this is growing in different colors, but mostly green. And it's, yes, produce penicillin. I will going to explain this briefly. And we run annotation of the genes, a prediction of enzymes, and looked on riboswitch. Riboswitch is a structured region of non-coding RNA, typically found within messenger RNA that acts as a sensor for specific molecules and upon binding regulate gene expression. Essentially, it's a genetic on-off switch controlled by the level of certain metabolites or other small molecules within the cell. And what I would like briefly to explain here is that the adaptation to high salinity of Dead Sea, it can be 60% or even more salinity done by riboswitch in this organism. So first of all, of course, assembly and assessment of genome was a question. We ran a few methods. Velvet, I cannot use, oh yes, no, I don't. Velvet spade soap, de novo method, and mazurka, abyss, and other methods. And as you can see, some predicted complete genes, many missing regions, fragmented regions, and at the end, the method was the most successful. Is the SPADA's genome assembly was best for downstream analysis and is the choice for future analysis, where we looked on predicted genes. And then a phylogenetic study classification based on a single copy orthologs was run, and statistically linkages were based on single copy orthologs for genome comparison using SPIDES assembly. And then we ran this phylogenetic genetic analysis, and we found that most of the Penicillium organisms are really closely associated with the our newly found organism. The organism was found to be distinct, not closely related to several industrial Penicillium strains. Next as a part was to annotate proteins, and this was done by mass spectrometry in our center. And bioactive synthesis and antibacterial activity was studied under using, first of all, E. coli and other bacteria. And proteome analysis workflow briefly was done using peptide file generation and processing, then sample preparation, transcript, and protein identification. In total, we identified more than 10,000 transcripts and annotated more than 7,000 different proteins, where more than about 5,300 were unique proteins found in this organism. Next, we looked on the synthetic biosynthetic pathway using anti-smash and validated by mass spectrometry to identify the production of penicillin in this organism. 89 of enzyme, their penicillin pathway production correlate to the original pathway identified with the validated peptides. But importantly, do the enzymes get involved into microbial biosynthesis pathway? The enzymes we identified, if indeed they produce anti-inflammation effect. So we ran a few experiments in different conditions, and we found that with high glucose and low nitrogen medium, it cannot produce any antibacterial effects, but on E. coli, using low glucose and high nitrogen, it produces antibacterial activity, meaning the penicillin molecules exist, but which type of penicillin we still need to identify. And then, as we know, this organism is stable for high salinity, pressure, and high temperature. And how can it be observed? We can look on many factors that can affect this tolerance, but could additional molecular genetic factor contribute to increase survivability of this organism? And then we looked on an interesting question. Could a riboswitch help for survival in the high salinity conditions? It can do upstream gene regulation, regulate gene expression and translation. It can do on and off target regulation, and make isomer switching and alternative splicing in eukaryotes. So we took all known isoforms in Penicillium species, and then compared predicted ribose switches in our organism, and we found identical and high conservation to THI ribose switch that regulate gene expression by binding to thiamine pyrophosphate, derivative of vitamin B1 in this organism. And actually, we predicted not just existence of this ribose switch, but also the secondary structure. And we found two of them. And in conclusion, just because of this brief talk, our results indicate the following. The discovery of a novel Penicillium species capable of growing under high salinity conditions, there's the potential to produce both secondary metabolites and novel enzyme activation. The presence of the conserved regulatory elements such as ribose switch, which may contribute to stress tolerance and overall survival. Expressionally variable strains that can produce secondary metabolites, this reduced dependence on fresh water resources. And current goals is to use this finding to newly identified genes will be explored for their potential in novel penicillin biosynthesis pathways, particularly under high salinity and high temperature. under climate change conditions. I would like to acknowledge my students, my collaborators, and funding, and I will be happy to have questions.
Thank you.
Thank you.
Unfortunately, there's no time for questions. If you want to ask a question, just talk to the speaker.
Thank you.
Let's thank the